Okay, now we're going to take a look at microevolution. This is generally evolution that occurs over a relatively small period of time. Geologic. The Earth is about 5 billion years old. Life started about 3.5 billion years ago. So when we say small amount of time, we're not talking 5 minutes, 10 minutes, 24 hours a week. We're talking a significant amount of time, but comparatively small. Okay? So that's because of the scale of evolution. So here's microevolution. It occurs relatively quickly versus macroevolution over a long period of geological time, over many, many generations. Okay, so here's some vocabulary that we need to know. The gene pool. Basically, the gene pool is if you take all the alleles in a certain population and you put them in a bucket, that's the gene pool. So all the big B's, all the big B, little b's, all the little b's, and you take all those individual alleles and you count them up. That would be the gene pool. Okay, so how many big B's are there? How many little b's are there? Allele frequency. This is actually counting and figuring out the percentage that those big B's contribute to the total population. Okay, so basically here on the left-hand side we've got three genotypes: big A, big A, big A, little A, little A, little A. So let's say you've got 50 people that are big A, big A. That means that those 50 people combined have a total of 100 big A alleles, because each of those 50 people have 2 big A's, okay? However, they have no little a alleles. This column here is big A, this column here is little a. Now, if we look at these people here, there are 40 individuals in this population, a group of people, that have big A, little a. That means they contribute 40 big A's and 40 little a's, so 40 and 40. Lastly, there are only 10 people that are little a, little a. They contribute no big A's to the population. They contribute a total of 20 little a, little a's. So if we add these up, 0, 40, 20, we get 60 little a's total. 100, 40, 0, 100 big A's total. The total of number of big A's and little a's is 200, which makes sense because we've got 100 individuals. Okay? And then to calculate the frequency of the alleles, we simply take the number of big A's and we divide it by the total number of alleles. So out of 200 alleles, 140 are big A's. That means that they have an allele frequency of 0.7, or 70% of the alleles are big A's. Okay? All right, now there are a couple forces of evolution. There are basically 4 things that come into play. One is mutation. So we've done mutation. Let's go back. So one is mutation. So we've done 2 different kinds of mutation: point mutations and frame shift mutations. Point mutations, there are 3 kinds. There are silent, in which case the amino acid doesn't change. So remember, a point mutation is one single base is getting swapped out. So if we swap out one single base and there's no change in the amino acid, because remember, there are in the amino acid chart, there are several ways you can form leucine. There are 4 different combinations of codons. Okay, so where were we? Okay, so point mutation, it can either be silent, in which there's no change, a missense, in which there is a change in the amino acid, and we don't know if that's good or bad, and a nonsense. Nobody likes nonsense mutations because they produce a premature stop in the sequence. Frame shift mutations, remember, you have an insertion in which you add a base, and the whole sequence gets changed after that. Or if you have a deletion in which you subtract the base. Remember the sentence: the big fat red fox ate the cat. If we insert a letter in there and we still read in groups of 3, because we read in groups of codons, that changes everything. If it's at the end of the sequence, it might not be so bad. If it's at the beginning of the sequence, it's really, really bad. Okay, so that was mutations. We're going to take a look in a couple of moments here about gene flow and genetic drift, and natural selection was in one of the other slide decks I made. Now, it's important to note that 3 things are random: mutation, gene flow, and genetic drift. Natural selection is not random. The only random part of natural selection is the mutation component of that. Okay? But mutation, gene flow, and genetic drift are random. Okay? So we've done enough with mutations. Mutations are not super, super common. Okay? They do produce variety in the gene pool, but they're not super common. There's got to be something else that drives evolution. Okay? Gene flow. So what gene flow is, it's when a population changes because individuals move out of the population and go into a different population. So here we have those purple beetles, okay? If one of those purple beetles leaves and joins a population of brown or tan beetles, it's called gene flow. We're changing the gene pool of the brown beetles. And now, depending on the type of inheritance, whether it's dominant or recessive, we might end up with purple or tan beetles. If it's incomplete dominance, you're going to have a blending of purple and tan beetles. I don't know what color that makes. Or if you have a codominant type of pattern, you're going to have purple and tan beetles, spotted or striped or something along those lines, okay? So that's gene flow. Genetic drift. Genetic drift can best be explained by, here's in the middle here, we've got a bottle of, let's say, all these different colored marbles. And if you turn the bottle upside down, only a couple of the marbles come out. You can think of it as a huge geologic event, a massive devastation of a population. It's called the bottleneck effect, because the population all of a sudden gets smaller and only the surviving members reproduce. So you have over here in this container a couple purple and one green. There are no orange. So that gene no longer exists in the population. That's what we call genetic drift. Another kind of genetic drift is what we call the founder effect. And the founder effect is when a few individuals start or found a new population, like, for example, the Amish population. And so that is going to be different from the populations that they left. The major issue with genetic drift and the founder effect is that if that gene pool stays stagnant, if there's no gene flow in or out of that gene pool, then the chances of having a heterozygous individual who is a carrier, unbeknownst to them, of a dominant recessive allele, there's a higher chance of two heterozygous individuals mating and then expressing one of their four kids will have the recessive disorder. So something along the lines of like sickle cell is an example of that, okay? So speaking of sickle cell anemia, over here on the left-hand side, we've got normal red blood cells. That's what it looks like. It looks like a jelly donut. Sickle cell anemia is when there's a problem with the hemoglobin. Hemoglobin's got 146 amino acids. And with sickle cell anemia, there is a mutation, and it's a point mutation, and it actually is a missense mutation. One single amino acid, off the top of my head, I think it's amino acid number 6 or 7, has switched. And that changes the structure of the protein that's produced by those 146 amino acids, which is hemoglobin. Red blood cells are coated with hemoglobin, and hemoglobin's got a couple jobs, but one of the big jobs it does is it carries oxygen. It cannot carry oxygen well if it's in a sickle shape. And additionally, these tend to stick together, and people with sickle cell anemia tend to form blood clots. Very painful and can be very, very dangerous. Okay, big S is the normal hemoglobin allele. Little s is the sickle cell allele. That means that homozygous dominant, you've got a normal phenotype. Heterozygous, you also have a normal phenotype, but you are a carrier. And if you're homozygous recessive, you do have the sickle cell phenotype, the sickle cell disorder, okay? Now, one of the things about sickle cell is if you have the normal phenotype, if you have big S, homozygous dominant, you don't have sickle cell, but you also do not have resistance to malaria, which is a mosquito-borne disease, okay? And malaria can be fatal if untreated, okay? Conversely, if you have sickle cell, your fitness is greatly reduced because of the sticky blood cells and also the reduced ability to carry oxygen. In the middle here, if you're a carrier, if you're heterozygous, you actually have the highest fitness because, number one, you don't have sickle cell, but you're also malaria resistant. Okay, so that brings us to the last portion of this. There are 3 different kinds of natural selection. We've got selection against the mean, selection against the extremes, and directional selection. So disruptive selection, phenotypes in the middle of the range are selected against. That means that the green line is before. That means that after the evolutionary process, we pretty much only have these phenotypes on the extremes that exist. The average phenotype doesn't exist. Over here, selection against the extremes, this is looks like what's happening with sickle cell, okay? So evolutionary-wise, we select against the guys on either end of the curve. Directional selection is we're pushing, like the stickleback fish. You're pushing away, you're pushing towards one particular characteristic, like having pelvic spines or not having pelvic spines. In the case of the sticklefish, it was not having pelvic spines in fresh water.